This video is going to go into the biochemistry of how hemoglobin is made, and I just want to start first by introducing this central dogma of biology. This just states that DNA moves to RNA to protein, and this was believed to be true in all things, but we know it's not true with retroviruses where RNA can actually go backwards and change DNA. Shown on this slide is a karyotype of 23 chromosome pairs, so a total of 46 chromosomes in all. You can imagine that the one on the left is coming from your mother and the one on your right is coming from your father, and this is being split apart in their gametes and passed on to the offspring. And while this occurs, multiple things can happen to include mutations that do not come from both parents but still occur randomly and spontaneously bring about changes. So two that I want to focus on is the 11th chromosome and the 16th chromosome. Both the HBB and HBA gene will contribute to creating two alpha or two beta chains, and those will come together to form a heterotetramer hemoglobin protein. And what's interesting about this is in the HBB gene around 7,000 years ago, there was a mutation that occurred on the 6th codon that changed the amino acid. And this property gave rise to sickle cell trait that would later cause sickle cell disease. And as we covered, the central dogma moves from DNA, RNA to protein. Now through gene therapy, clinical trials are allowing people to go into the second chromosome where the BCL11A gene is, and they can change activity in this gene that affects the expression of genes and can actually reverse time on the HBB gene. So instead of producing adult hemoglobin beta chains, it's pulled back to its fetal hemoglobin production that can cure or alleviate problems associated with sickle cell disease, or that's what's being seen in clinical trials thus far. So what I was just covering is this HBB gene. This is the location on it. It creates this beta chain, but as you can see just to the left of it is this delta sign for fetal hemoglobin. And this is going to produce the pink strands that we see here. So it'll form a dimer of beta chains. And then here on the HBA from the 16th chromosome, this is going to create the dimer of alpha chains. And since these are two different chains coming together, it's going to form a heterotetramer with 4 subunits that's known to be in hemoglobin. So just to be clear with this too, the red blood cell contains the hemoglobin. That's what we're interested in looking at. And each hemoglobin chain contains one prosthetic heme group within it. On this slide are the 20 amino acids, and they are grouped based on their properties. The nonpolar are more hydrophobic and tend to be facing inward, while the polar groups that have charges on them or hydroxyl groups that can interact with the environment, they face outward. And also the opposite charges will interact with each other. So if you have a positive charge facing outward on one chain and a negative charge on another chain, these two can actually interact and give special properties to proteins. So just one example of how this occurs is histidine, which is shown up here in the top left, and on the bottom is a titration curve that shows how it either gains or loses hydrogens based on the pH of the solution it is within. And on the right side, we can see where this occurs in the F8 region of hemoglobin. And what it does is the histidine will modulate the oxygen binding and releasing from the iron, and this will create either a planar structure as shown when oxygenated, or it will fold and become nonpolar when deoxygenated. And this will allow for when oxygen comes back to it, it will be attracted to it and pull it inward again and resume the oxygenated position. And histidine is of more interest inside the body because the pH is around 7.4, and that's close to where the PI value is for it. So amino acids are linked together to make polypeptide proteins. This is a condensation, or you can think of it as a dehydration reaction where H2O is released and a peptide bond forms between the carboxylic group and the amide group. So levels of protein structure, they fold to make globular proteins, or they can form other unique structures. In this case, the image below matches well with what is being made in hemoglobin. We've already covered some. The primary structure is laid down by amino acids that are linked in a long chain. The R groups of these will determine how it folds and how they interact. Those will give rise to the secondary structure, which can form alpha helices or beta sheets. In the case of the globin family and hemoglobin, we're just going to produce alpha helices, and I believe it's going to be 8 conserved across the globin family. So these can form tetramer units such as myoglobin, and this will contain within it a peptide chain of a certain length, and it's also going to have secondary structures formed such as the 8 alpha helices. And then finally, the quaternary structure gives rise to the heterotetramer hemoglobin, which is the largest of the globin family and has 4 subunits that can interact with each other to allow cooperative oxygen binding. So the beta chain primary sequence is laid out below here. We show that it's the beta chain with the symbol, and A just is the average adult normal wild type. And here we have these amino acids running across, and we can see that the properties start to change. Once we get to 6 and 7, they start to become polar. So we could imagine these are going to be facing outward and interacting with the environment. And the sickle cell mutation, the 6th position here, is actually mutated, and it causes the amino acid substitution from this polar glutamate to a non-polar valine, which is generally hydrophobic. And this occurs in a position where when the red blood cell is opened or closing based on its conformational changes, if it has oxygen or not, then it allows these mutated areas to aggregate together and disrupt functions at higher levels. So the HbS is the most common seen in sickle cell disease, but there's also HbC, which is a variant where instead of valine being produced, lysine is produced. And this is also the 2nd most common in Africa, where someone inherits the HbS allele that arose 7,000 years ago and later inherited the HbC allele that came along at some point. So myoglobin and hemoglobin both have very similar secondary structures. Here on the left, we can see where it's lined up in primary sequences with a few areas skipped. And you can see that there's a lot of commonalities among the primary structure. And even if there are differences, there's still similarities in the properties of the substituted amino acids. And that gives us these similar structures that we see on the right side here. So still same amount of alpha helices and still containing a heme group. So globin proteins share tertiary structures. You can see with this overlay how all of these structures look very similar to each other, although there are some variations. And this is because the percent identity is similar for the most part, where 28% of myoglobin matches the alpha hemoglobin, which is inside the 2 chains of hemoglobin. And it's also very close to these other monomeric units as well. So hemoglobin goes a step beyond these monomeric units, and it actually has a quaternary structure. This gives it special abilities where it can cooperatively bind oxygen and move it from the lungs, where it's oxygen rich coming in, to tissues that need oxygen, and it can unload it. And this is accomplished by these two chains, the 2 alpha and 2 beta chains, which are 50% identical. And so they're very related to one another, and they interact with each other in a way that allows oxygen to be transported in a quaternary fashion that could not be accomplished at a monomeric level. So hemoglobin has 2 conformations, the T state, which stands for 10th state, and also the R state. And it's from switching from these 2 states, which is what allows the oxygen to cooperatively bind or not. And also with the mutations of sickle cell disease, in the deoxy state, we can imagine that the valine is pressed outward more than it should be, and that's what's given rise to the problem with cell sickling in deoxygenated states. So here is the O2 ligand binding curve. The myoglobin, we can see on the left, just strictly binds the oxygen and holds on to it until it's needed, and then it can be released. Whereas hemoglobin has properties where if it binds one, it promotes the other regions to also bind oxygen, and same for unloading oxygen once it reaches tissue. Wild-type HbA and HbB genes will make adult hemoglobin, which is commonly just referred to as HbA. But since it's inherited from both parents for you to have the disease, I think it's easier to see it as HbAA or HbS. So the first person or carriers of sickle cell trait would only have this HbS variation. So they would have one spot where there would be a valine in the position where it's not supposed to be, but this would not cause problems except in severe situations. So working out or climbing a mountain and not having much oxygen available while being dehydrated. So in those circumstances, the cells will sickle, but overall there will be no problem, no aggregation. However, if someone inherits 2 HbB mutations, so one from each parent, then they will have HbSS hemoglobin, and this will cause the mutant amino acids to aggregate together. And what happens is you can see this beta chain here, and this beta chain both have valines in positions that they're not supposed to, and whenever these come in contact with additional hemoglobin, those will interact with other beta chains in a negative way that will form aggregation. And I'm jumping ahead of myself here, but this delta, this is what they're trying to pull back gene therapy to. So instead of going down this timeline and becoming an adult and producing beta chains, by changing the expression and altering the chromosome 2 HCl11A position, we're able to stimulate this fetal hemoglobin, and this beta hemoglobin can't provide any issues anymore. To understand how mutant amino acids cause red blood cells to sickle and to sickle cells, I relied a lot on this textbook, Sickle Cell Disease. This was released in 2010, and you can see the publishers here. This is a really great book for allowing me to get more insights in how sickle cell disease manifests in a molecular way in the body and gives rise to many of the problems associated with the disease. So these figures here were really useful and I think explain it pretty well. The adult hemoglobin consists of usually 2 alpha chains and 2 beta chains, and then if no mutations occur, as in most people, we have this right here, and this is normal. But in the case of inheriting 2 mutated alleles, then we produce 2 valines in positions where we're not supposed to, and in an oxygenated state, the valine is of no problem. Everything continues as normal. But in the absence of oxygen, the conformational change allows valine to stick out more so. And this interaction of valine with other regions is what causes polymerization. And on the right side, we can see here how these molecules are generally free floating, and they will actually start to link up together and form these really long chains. And what you see is on the inside, all of these chains become linked and make really long aggregated lines. And after a long time, these do not come undone from each other, and it presents this outward sickle appearance. And I think this slide does a great job too, or this figure does a great job covering the deoxy HbS polymerization. These circles we can imagine as the alpha 1, alpha 2, beta 1, beta 2, and it's showing that these valines at the 6th position, which we know is where the mutation occurs, it actually sticks out and will get stuck in these regions here of the other beta chain. So we'll have linking here, linking here, linking here, and all of those are problems that arise because there's no longer this glutamic acid sticking out and preventing this from occurring, whereas this valine is now attracted to these nonpolar regions and it's causing issues to arise. So that covers the aspects of the biochemistry of building the hemoglobin from its primary structure to its tertiary structure, and how the mutations can interact to cause aggregation throughout the body. And what I want to focus on next is how the molecular mechanisms that cause these aggregations result in physiological things that can be observed in the body and give rise to the disease and make people actually go to the hospital and get treatment, and is really driving the work behind genetics and different therapies in order to cure this disease.